Hi, my name is Katrina McLeod and I'm a scientist at IMAS, and I'm going to give you an overview of some of the student projects that were involved with the MOMA Heavy Metals project in 2016. In 2016 we had 4 students working on research in the Derwent supported by scholarships from MOMA. They also worked alongside the Derwent estuary programme and a variety of other industries within the link to the Derwent system. The 4 students were Ada Crantock, and Ada's project looked at Pacific oysters as a monitoring tool. Trung was interested in whether we could use remote sensing data to help improve monitoring in the Derwent. Kate was also looking at monitoring, in this case whether we could use epiphyte communities. In actual fact, she looked at epiphyte and epifauna. So these are the plants and animals that grow on surfaces, and she wanted to know whether we could use them to reflect different levels of nutrient load. And finally, Madalyn Riley. Maddie was looking at the effect of heavy metals on little penguins, and she's still going. Maddie's project's not quite finished yet. So I'll run through each of those projects now. Firstly, Ada. As I say, Ada was looking at improving approaches for using oysters as a monitoring tool. Oysters are already used in the Derwent estuary through the Derwent estuary programme and the near-stir monitoring, where they deploy them and use them as a proxy for metal uptake. But what we wanted to look at more particularly, or what Ada wanted to look at, was whether the size of the oyster influences the metal uptake, whether the oyster deployment time would have an effect on metal uptake, and also whether we could use the shell as a proxy for flesh load. So that usually we're interested in the meat of the oyster because that's the bit people eat. But if for some reason the oyster dies or we lose it, that flesh load, we wondered if perhaps the shell could be used in place. Ada had 3 experiments that she undertook: the field experiments, lab experiments and deployments. So the first of those was an oyster deployment where she put out a large number of oysters for a period of 10 weeks, and we used this to determine what is the optimal deployment time. The second experiment collected oysters from a range of sites around the Derwent to look at what is the spatial coverage that you need. And then the third one used shells from the wild oyster deployment to look at the correlation between the levels in the flesh and the shell, and that was using a technique called laser ablation. What were her key findings? Well, firstly, the size does not change the metal load, size of the oysters. Therefore, using small oysters is actually a more cost-effective option. They're easier to deploy and cheaper to obtain. Ada found you had to have them out for at least 5 weeks in order to get a really good measure of the variability in the environmental load. And unfortunately, the shell levels did not relate to the loads in the meat, and therefore that's not an option for the future. There were quite strong regional differences within the estuary, so yes, we still need to have quite an extensive deployment programme or collection programme to get a good understanding of that regional variability. Our second student, Trung, looked at the spatial data and how we can use that to inform monitoring. Trung really looked at remote sensing data. So the first thing we looked at was what data was available for the Derwent, and then secondly, how reliable is that remote sensing data? Can we relate it to real observations and be confident that it's telling us what's happening out there in the system? And then finally, he had a look at whether some of that data could be used through modelling to actually give us an understanding about particular problems. And in this case, he used POMS, Pacific Oyster Mortality Syndrome, as a test case. You might be aware that POMS had a major effect last summer on our commercial oyster industry, devastating it with large-scale mortalities. So the view was if we could use some of the monitoring in the Derwent to give people a heads up if there was a risk of POMS at any given time. So looking at the remote sensing data, we found some quite good coverage in relation to CDOM, that's the coloured dissolved organic matter in the system. In relation to chlorophyll, there was good data. There was also good data on turbidity and temperature. And the temperature one is particularly interesting because many biological processes are related to temperature. When we compared the remote sensing data, so the satellite data, with real-time information, so field observations, the correlations were pretty poor. The best we had was with the temperature data. But that was largely because of the mismatch between the satellite timings and the field timings. So they weren't done on the same days. And that's something we could look to rectify into the future. So by a little bit of forward planning, we can try and make sure that we try and get our field observations at times when satellites are in the area. Trung's model was proved to be quite useful, classifying the main areas within the systems, so these red and orange areas, as those that are hotspots for detection of POMS. And so the key findings from this project were that we need to improve our timing with relation to field data and the remote sensing data, but that the modelling can help identify that. The remote sensing data can inform the models, and the models can inform environmental assessments for POMS. And the key areas here were Ralph's Bay and the lower estuary, if you were looking to focus monitoring. Kate looked at the epibiota and whether they can be used as indicators of nutrient change. The key questions here were whether we could first off get them to grow under different nutrient conditions, and largely we focused on epiphytes here, so the plants, and that was to the proof of concept that the idea would work, and then we took it to the field. Taking it to the field, the next question was what sort of deployment strategy would work best? How would we get them out there? And then the third was once you've pulled the data back in, what is the change that we're looking at? Is it changes in the animals and the plants that are there, the actual types of animals and plants, or is it just their abundance and their size? So that was the third question Kate looked to answer. The first experiment was the testing the nutrient gradient, and here she put plates which we had seeded with little plants, little algae, into 3 different concentrations of nutrients, and results showed quite a clear difference with plants growing best in the high nutrients. The next point was looking at spatial variability with field trials, so we put these plates out, but we also put out other substrates that we felt might mimic the kind of habitats you'd find in nature. So we put out loofahs, which mimic the complex habitats associated with sponges and weeds and seaweed areas, reef areas. We put out pipes, very common. They represent crevices, but also quite commonly flat surfaces deployed in harbours, marinas, fish farms, and similarly ropes. They're a very common habitat in our marine systems these days. What Kate found was that the pipes, loofahs and ropes all were promising. They all showed a gradient in response, largely in relation to the amount of species rather than the particular types. There weren't major changes in types. Unfortunately, the pipes were amongst the most promising, because whilst the loofahs and the ropes were great, they were really difficult to get the little animals and plants out of. So yeah, pipes definitely look like they could be a useful tool for epiphyte and epifaunal monitoring into the future. Maddie her project is still underway. She's still analysing the last of her data, but her main project was to look at whether penguins in the Derwents might be affected by heavy metal contamination. She did this by looking at metal levels in moulted penguin feathers, and she collected these from a variety of penguin rookeries around the Derwent and surrounds, and she is looking at whether metal levels vary between locations, and also whether there might be any evidence of bioaccumulation as a result of the different food sources. So if the locations vary, is that because there's different loadings in those areas, or is it because the penguins are eating slightly different things? So that's a very exciting study, and look forward to seeing Maddie's results when they get out. Watch this space. I've included the IMAS website there, so that and the Derwent Estuary Programme, Penguin Programme, will certainly highlight Maddie's results when they come out. So finally, I'd just like to really thank Mona for their heavy metal scholarships and investing in these studies in this science. The project looks to bring together art and science with a view to getting people to look at the pollution problem in the Derwent from a range of different perspectives. And, you know, thinking a little differently about it often can help us solve a problem. So thank you to Mona, and I hope you've enjoyed this little talk and the rest of the presentations, demonstrations, and the Mona market. Thank you.